Is everything okay? Do you hear me? Do you see my screen? Okay. So, hello everyone. Thank you for having me. I'm Parmida Pezeshki on behalf of OncoDam team, and I'm going to present our Impact Scholars project on how image segmentation affects classification implications in a cancer diagnostic model. So the aim of our project was to see how image segmentation can improve or affect the classification accuracy on histopathology data sets from cancer tissues. So first, let's talk about image segmentation a little bit. Image segmentation is the process of dividing an image into distinct parts or regions, and it can make analysis or some interpretations easier. Each segmented image can correspond to a specific object, feature, or an area of interest in the image. Image segmentation can have applications in medical imaging. As you can see here, this figure is from MedSam paper. Image segmentation can help in medical imaging. Segmentation can help identify and isolate, like for example, organs, like tumors or tissues, and it has been very helpful in performing these tasks. There are several methods to perform segmentation, such as thresholding, edge detection, region-based, but advanced deep learning models, such as Meta AI's SAM, or Segment Anything model, are the state-of-the-art models for this task. So for our project, we used two data sets. The first one was the breast cancer histology images, or BAC data set. This data set included breast tissue images in four subclasses of normal breast tissue, benign tumors, in situ tumors, and invasive tumors. This data set did not have any ground truth segmentation masks provided by the authors. So for this data set, we had to generate the segmentation masks, using segmentation models such as SAM or MedSAM. And the second data set was DigestPath, or Digestive System Pathological Detection and Segmentation data set. This data set included colon tissue histopathological images in two subclasses of negative images or normal colon tissue, or positive images, which included some parts of colon tumor or colon cancer parts. And in this data set, the authors provided ground truth masks. So we had these ground truth masks provided by the authors to showcase that regions of colon tumor. And the ground truth masks were only provided for positive or tumor images, because the normal or negative images only constituted the background normal colon tissue. So here is our study pipeline for training the model. We used two training pipelines. For the first half of experiments, we used original unsegmented histopathology images as our input, and then we preprocessed these images by resizing and transforming the images. And then we fine-tuned and trained our classification model, which was Google Vision Transformer, or Google VIT, and obtained accuracy measures. For the second half of the experiments, we again used histopathological images from both data sets as our inputs. But here we first segmented the images using generated masks for back data set. As I said, the data set didn't come with ground truth provided masks. Or we segmented the images using ground truth masks for the DigestPath data set. So pre-processing here included applying the masks to create the segmented image, and then resizing and transforming the image. And then we also fine-tuned and trained our Google VIT model on these images, and obtained the accuracy and compared the accuracy of these like different pipelines. You can see our experimental design here. Overall, we run four kinds of experiments. As I said, like the training was done either on unsegmented original images or segmented images. And also the testing was done on either unsegmented or segmented images. But the main objective was to like compare the classification accuracy when training and testing on both, like when both training and testing is done on unsegmented original images, and when the training is done on segmented images, and then the validation and testing is done on unsegmented original images. Because the idea was that like training on segmented images might like focus the model on that areas of interest and make the model learn faster where to look at to like label the images correctly. And for all the experiments, the variables such as number of epochs, learning rates, and other variables were fixed and were similar. So as I said, we had like two options for generating the masks, SAM and MedSAM. So we first had to compare the segmentation accuracy of these two models. And because we had the ground truth masks only for the DigestPath dataset, we used this dataset to like obtain the segmentation accuracy of SAM and MedSAM on this kind of this type of dataset, which is like cancer histopathological images. And MedSAM is a variant of SAM, which was fine-tuned on multiple medical imaging modality images like CT scan, MRI, and also like histopathological images. But if you read the paper, the histopathological images include a very small part of their training data set. And surprisingly, like SAM performed better in segmenting these histopathological images. The segmentation accuracy for SAM was about 64%, but for MedSAM, it was about 33%. So we continued with SAM and used it to generate segmentation masks when the dataset didn't come with ground truth segmentation masks. So let's talk about our results or observations. Observation number one was that segmentation does not always improve accuracy. For the back data set, training with segmented images and then testing on unsegmented images resulted in an increase in the classification accuracy compared to when training and testing both were done on unsegmented original images. But for DigestPath dataset, where the segmentation masks were 100% accurate and like they were ground truth masks, when we trained the model on segmented images and then tested on original unsegmented images, the classification accuracy dropped by about 20%. So we hypothesized that this observation could be first because 100% accurate masks would omit all the background noise completely. So the model is very prone to overfitting. And the second hypothesis was that, and it was one of our main problems, as I said, the segmentation masks were only provided for the positive or tumor images because the negative images were only like normal background colon tissue. So the classifier apparently could rely on the presence of this segmentation mask in positive images in training as a cue. So when tested on unsegmented images that lacked this cue, which was the segmentation mask, the model tended to misclassify almost all of the test images as negative. So observation number two was that, like one of our other hypotheses was that, like if we change this dimming factor, because we used a dimming factor to like dim the background and highlight that region of interest, and like we wanted to see if we change this dimming background and like increase it then the background noise would probably decrease, and it would affect the accuracy, classification accuracy, but like changing the contrast and the dimming factor had minimal impact on the accuracy of the classifier. The third observation was related to the digest path dataset. So as I said, when we trained the model on segmented images, the accuracy for digest path datasets dropped significantly. And we hypothesized that this was because like the background noise was almost completely omitted in this dataset. So when we trained the model on a mixed dataset of both segmented and unsegmented images, like it was kind of a duplicate datasets. It included both like segmented and unsegmented images. The accuracy improved significantly. And it was almost like the same as when trained and tested on unsegmented images. So for our conclusion, we can say that segmentation does not always improve classification. And in some cases, it can also hinder generalization. When highly accurate masks are used, they may cause overfitting because they reduce the performance on tests on segmented images by omitting the background noise. But lower accuracy masks can improve generalization by retaining useful background information while also making the model focus on the regions of interest. And segmentation, but last but not least, we can say that segmentation effectiveness depends on various factors such as application and mask quality, and it requires further research and evaluation to like find out more about this effect on classification accuracy. And talking about our limitations, the limited publicly available histopathological data sets was one of the setbacks in this project. We actually spent a considerable amount of time like finding histopathological data sets which were suitable for both a classification and segmentation task. And we can say that there were not a lot of them available online and publicly. So we wanted to point out the importance that like medical institutions that produced this highly valuable data sets care and value more about like publicly publishing these data sets. So the research in this field can move forward faster. We would like to thank the Neuromatch Academy and the Impact Scholars Program on behalf of our team that you can see here. And the codes of this project are available at this GitHub repository if you were interested. Thank you for your attention. And if you have any questions, I'd be happy to answer.